Good morning, everyone. We are the Group 11, which we will tackle about the extension of Mendel's two-gene inheritance. Extension of Mendel's two-gene inheritance. Learning objectives. First, conclude from the result of crosses whether a single gene or two genes control a trait. Second, infer from the results of crosses from the existence of interactions between alleles of different genes including additivity, epistasis, redundancy, and complementation. In the chapter, opening additive interactions between two genes controlling a single trait can result in novel phenotypes. When two genes contribute additively to a trait, their combined effect is greater than the sum of their individual effects. This can lead to the emergence of unique phenotype characteristics that are not simply a blend of the parental traits. These interactions are essential in understanding the complexity of genetic inheritance and the diversity of traits observed in populations. The inheritance of traits from parents to offspring can be more complex than simple Mendelian inheritance, which involves one gene controlling one trait. When we talk about two-gene inheritance, we're likely referring to the interaction of two genes in determining a particular trait. This can be explained through a concept known as the hybrid inheritance. The hybrid inheritance. First is the hybrid cross. In a dihybrid cross, you examine the inheritance of two different traits controlled by two separate genes. Each gene has multiple alleles. Alleles are different forms of a gene that can encode for variations of a trait. For each gene, an individual inherits one allele from each parent. Independent assortment. One of the fundamental principles of the hybrid inheritance is the law of independent assortment, which means that alleles of different genes segregated independently during gamete formation. This results in a variety of possible combinations. Fourth is the Punnett square. This is to predict the offspring phenotypes. A Punnett square is often used, which helps show the possible allele combinations resulting from the combination of alleles from the two parents. The phenotypic ratios. The phenotypes of offspring can vary depending on which alleles they inherit. Different combinations can result in a range of traits, leading to different phenotypic ratios among the offspring. How two genes interact to produce seed color in lentils. The inheritance of seed color in lentils, like in many other organisms, is often influenced by the interaction of multiple genes. In lentils, the genes responsible for seed color are typically controlled by multiple loci, specific positions on chromosomes, leading to a polygenic inheritance pattern. Here is a simplified explanation of how these genes interact. First, the multiple genes. Seed color in lentils is determined by more than one gene. Let's denote these genes as gene A, gene B, and so on. Each gene can have different alleles versions, and the combination of alleles at this gene's loci determines the final seed color. Allelic combinations. Different combinations of alleles at this gene loci can result in various seed colors. For example, if gene A has alleles for brown seed color AB and gene B has alleles for green seed colors AAB, the lentil seed will likely be brown. This epistasis. An interaction. There can also be epistatic interactions where the alleles at one gene locus can influence or mask the expression of alleles at another locus. This interaction can lead to unexpected seed colors. For instance, an allele at gene A might mask the expression of gene B, resulting in a different seed color than what is predicted by individual gene combination. Another is the environmental factors. Environmental factors such as light and temperature can also influence seed color expression. These factors can modify the phenotypic outcome, even with a particular genetic combination. So, in summary, seed color in lentils is a complex trait controlled by multiple genes with various alleles and possible epistatic interactions. The final seed color is determined by the specific combination of alleles at this gene loci and can be influenced by environmental conditions. This complexity can result in a wide range of seed colors, contributing to the diversity of lentil varieties. This extension of Mendel's principles allows us to understand how multiple genes interact to determine a specific trait, taking into account the complexity of genetics beyond simple single gene inheritance.
I am Mr. Basin Lab. I am going to discuss the figure 11 and 12 in chapter 3. Epistasis, alleles of one gene can mask the phenotypic effects of alleles of another gene. Epistasis is a genetic phenomenon where alleles of one gene can mask the phenotypic effects of alleles of another gene. In the context of lentil seed color inheritance, two genes denotes as big letter A and big letter B control the production of enzymes responsible for pigmentation. Allele A encodes enzymes A and contributes to the formation of tan pigment. The allele B encodes enzymes B responsible for gray pigment. The presence or absence of these enzymes, along with the interaction between the genes, determines the seed's color. In case in Labrador retrievers, coat color inheritance is governed by two genes, which is the E genes, where determines the presence of dark pigment or eomelanin, and the absence of light pigments, which is pheomelanin, and the B gene, responsible for depositing eomelanin in the hair. The presence of the dominant E allele ensures the production of black color, while the recessive E genotype result in yellow color due to the synthesis of only pheomelanin. Additionally, the B genes comes into play. The B allele ensures efficient eomelanin synthesis and deposition, leading the black color, whereas the small b allele is less efficient, resulting to the chocolate brown color. When a Labrador retriever is homogeneous, recessive for the E allele, the double A will result. It masks the effects that alleles at the B genes, causing the dog to appear yellow regardless on the B allele's presence or the 4S. This phenomenon, known as recessive epistasis, demonstrates how the presence of certain alleles can hide the effects of alleles and another gene, leading to specific coat color outcomes in the Labrador retrievers. The interaction between these genes creates different phenotypic classes, including black, chocolate, and yellow coat colors. That's all. Thank you.
Good day, sir. I am Michaela Denise Bantilan, and I am assigned to explain the figure 3.13, the biochemical explanation for cost color in Labrador retrievers. So, protein E activates an enzyme that generates eumelanin from a colorless precursor. When protein E is present, only eumelanin is produced. Protein B deposits eumelanin densely so that the hair is black. So if eumelanin is dense, the color of the Labrador will be dense to black. If the eumelanin is not dense, it will result to brown hair or chocolate. Protein B deposits eumelanin densely so that the hair is black. Pigment is deposited less densely without protein B, producing brown hair or chocolate. So in the absence of protein E, no eumelanin is produced, and instead pheomelanin or the yellow pigment is synthesized. Homogeneous E dogs are always yellow regardless of the gen B genotype. The reason is that protein B affects eumelanin only, but these E animals have no eumelanin. So the reason for homozygous E-dogs to be always in yellow regardless of their Gen B genotype is because uh, the protein B affects eumelanin only, and the homozygous E-dogs don't have eumelanin, instead they have pheomelanin. So that is the reason why homozygous E-dogs are only of yellow color, not in black or in brown or chocolate shade.
Good day, sir. My name is Rachel Maata Marchand, and today I will going to report my assigned topic, which is recessive epistasis in human blood types. So first, recessive epistasis, it occurs if the recessive allele of one gene hides the expression of all alleles of another gene. So, recessive epistasis in humans causes a rare blood type. A homozygosity for the H. bombi allele is epectatic to the I gene, determining A, B, O blood types. HH individuals fail to produce substance H, which is needed for the addition of A or B sugars at the surface of red blood cells. Because H is epistatic to I. Rare individuals may appear to have blood type O despite of having an A to I of I to B allele. When the masked I allele is expressed in their H progeny, these people may be surprised by the child's blood type. So for letter A, molecular basis of the Bombi phenotype. So Bombi phenotype is mutant recessive allele at a second gen HH mask phenotype at ABO alleles. Well, how many is epistatic causes unexpected inheritance pattern of ABO blood type?
Good day, sir. This is Barbie J. Salak, and I am the next reporter. My topic is all about predicted F2 ratio, genotype, phenotype, and final ratio. So let's start with ratio. In genetics, a ratio refers to numerical relationships between different traits or alleles. It helps us understand how certain traits are inherited and how they might appear in a population. Ratios can be expressed in various ways, such as 3 ratio of 16, 9 ratio of 16, 3 ratio of 16, and 1 ratio of 16, depending on the specific inheritance pattern. Next, let's talk about genotype. Genotype refers to the genetic makeup of an individual organisms. It represents the specific combination of alleles, of version of genes that an individual inherits from its parents. Genotypes can be represented using letters such as big letter A underscore 2 small letter B, big letter A underscore big letter B underscore 2 small letter A and big letter B, and 2 small letter A and 2 big letter B, where uppercase letters typically represent dominant alleles and lowercase letters represent recessive alleles. Moving on to phenotype. Phenotype refers to the observable characteristics or traits of an individual or organisms. It is influenced by both the genotype and the environment. Phenotype can include physical traits like eye color, height, or hair texture, as well as behavioral traits like intelligence or temperament. Phenotype can vary even if individuals have the same genotype due to factors like gene expression or environment influences. Lastly, let's discuss the final ratio. The final ratio is the expected or observed ratio of phenotype resulting from a genetic cross or breeding experiment. It is determined by the combination of genotypes involved, the inheritance pattern of the specific traits being studied. By analyzing the final ratio, geneticists can gain insights into underlying genetic principles governing inheritance and make predictions about the likelihood of certain traits appearing in future generations. That's all for my topic. Thank you.
So, good day everyone. Good day, sir. I am Agnes Blair Tisanas and I am the reporter for the Figure 3.6. Biochemical explanation of reciprocal recessive epistatus of the generation of a sweet pea color. So, enzymes specified by the dominant alleles of two genes are both necessary to produce pigment. Recessive alleles are both specify no enzymes. In AA homozygotes, no intermediate precursor 2 is generated. So even if enzymes B is available, it cannot produce purple pigment. So because it takes two enzymes catalyzing sequential biochemical reaction to change a colorless precursor into a purple pigment. Only the A negative, B negative genotypic class, which produces active forms of both required enzymes can generate colored flowers. So the other three genotypic classes like A negative, BB, AB negative, and AABB become grouped together with respect to phenotype because they do not specify functional forms of one or the other requisite enzymes and thus give rise to no color, which is the same as white. So it is easy to see how the 7 part of the 9:7 ratio encompasses the 3:3:1 ratio of the 9:3:1 F2 ratio. So the 9:7 ratio is the phenotypic signature of the type of reciprocal recessive epistasis, in which a dominant alleles of two genes acting together, A negative, B negative genotypic classes, and A negative BB, AB negative, and AABB. So, given that the phenotype associated with the either allele A or allele B is purple, then we can say that the AA is asymptotic to B and BB is asymptotic to A. If the sweet peas are either AA or BB, their flower will be white regardless of whether or not they have dominant allele on the other gene. So the two genes work in a tandem to produce purple pigment. That's all and thank you.
3.7 Dominant epistasis may result in a phenotypic ratio based on the picture below in letter A. In summer squash, the dominant B allele causes white color and is sufficient to mask the effect of any combination of A and A alleles. As a result, yellow A negative or green AA color is expressed only in BB individuals. For the letter B, the A allele encodes enzymes A, while the A allele specifies no enzyme. Therefore, yellow pigment is present in A squash and a green pigment in A squash. A deposition of either pigment depends on a protein B encoded by allele B, the normal white type allele of A's second gene. However, the mutant dominant allele B encodes an abnormal version B of this protein that prevents pigment position, even when the normal protein B is present. Therefore, in order to be colored, the squash must have protein B but not protein B or the genotype BB.